Good morning, everyone. Thank you very much for the kind introduction. I'm Priyanka Ghosh, and I'm very excited to talk to you today about an emerging technology that we are currently exploring to develop alternative BE approaches for topical dermatological products. Specifically, I'm going to talk to you today about non-invasive Raman spectroscopy-based bioequivalence approaches. The learning objectives of this presentation today are to understand what non-invasive imaging-based techniques are and how we can utilize these techniques in generic drug development, specifically to better understand the product microstructure, to understand metamorphosis of the dosage form and drug release, and for evaluation of cutaneous pharmacokinetics. Now, before I get into that, this is a very simplistic representation of how we obtain a Raman signal. So you basically have a laser which you focus on the sample and you obtain a spectrum using a detector which is very comparable to a HPLC spectrum, for example. You can use the area under, say, for example, a peak of interest or the height of the peak of interest then to quantify or detect your molecule of interest in any given sample. Now, this is not a new technique in the pharmaceutical industry. Raman-based techniques are routinely used and are a very prominent uh, tool within the analytical toolkit where they are utilized for identification of active ingredient, of degradation products, of isomers, polymers when they exist, of fresh contaminants, as well as of inactive ingredients. So why are we talking to you about this today then? We are talking to you because we see an incredible opportunity ahead of us where this technique can be utilized for developing more efficient bioequivalence strategies, specifically focusing on topical dermatological products for this presentation today. So where do we see the potential utility? We see an utility in better understanding the dosage form. This is an example from the literature where the authors used hyperspectral imaging techniques to understand the relative distribution of three APIs shown here in pink, in green, and in blue, as well as an inactive ingredient shown here in yellow. Now, how do we utilize this in, say, for example, for a topical dermatological products? Imagine if you are able to understand the relative distribution of an API in the continuous phase versus the dispersed phase in an emulsion formulation. That would enable us to better compare and contrast the microstructure and develop more efficient characterization-based approaches. The second example that I have here is to look at drug release. The authors utilized a Raman-based strategy to look at the amount of carbamazepine in a release media over time. And because they use a Raman-based technique, they were able to detect not only the amorphous material, but also the relative distribution of the dihydrate and form 1 and form 2. Thinking about topical dermatological a product, if I'm thinking about release, I'm envisioning that if I'm able to track an API and an excipient simultaneously over time using a Raman-based techniques, I can not only utilize it to understand release from the drug product, I can potentially also use it to understand the metamorphosis of the formulation on the surface of the skin over time. The last thing that I would like to show you, again an example from literature, is where Raman can be utilized to evaluate drug disposition. What do I mean by that? In this example, the authors utilized Raman to understand the relative distribution of ketoprofen, the API in this example, across different depths of the skin as well as over time. And not only did they look at the API, they also looked at the relative distribution of an excipient propylene glycol in this example. And this provides us with an invaluable tool where we can use techniques like Raman to look at the local bioavailability of the API at or near the site of action in the skin, and therefore utilize this technique for evaluation of bioequivalence. Now we have begun to explore some of these techniques. The first example that I'm going to show you is related to product microstructure within our GDUFA Regulatory Science Research Program. Now, in this example, we looked at bioavailability of acyclovir from, again, these Zovirax products, product dispensed using a tube, compared to the product dispensed using a pump. These products are available in the UK, and the data showed us that there were differences in the bioavailability of acyclovir when the product was dispensed using these two container closure systems. Now, what did we do next? We looked at the microstructure of the product. And what you are looking at here is a representation of Raman images. We looked at the product in the tube. We also looked at the product from inside the pump, and we compared it to the microstructure of the product that we observed for the product that was dispensed using the pump, or in other words, the product that experienced the shear stress. We saw these dimethicone globules in the products that were dispensed using the pump, which we were then able to correlate with the performance of the product, and this entire data set enabled us to better understand the microstructure of the product, better understand the correlation between the microstructure and product performance, and also enabled us to institute adequate controls. Say, for example, if a generic manufacturer would like to manufacture a bulk product and package it into two container closure systems, it may be necessary to have additional controls in place to ensure that the microstructure of the dispensed product is comparable to the microstructure of the bulk product, and therefore the bioavailability would also be expected to be comparable. Next example that I have for you is from the literature. In this example, the authors used Raman-based techniques to look at the deposition as well as the metamorphosis of the dosage form. In this case, we are looking at ibuprofen. So the authors looked at the formation of these ibuprofen crystals on the top of the skin following drug deposition. Now, in the lateral view, you can of course look at the deposition of the ibuprofen on the surface of the skin. You can also look at the distribution of the propylene glycol, which happened to be the vehicle in this case. And one additional example that I think is important for me to point out, because the authors use simulated Raman scattering techniques, which is more powerful than inherent like confocal Raman, they were able to detect the molecule without any tagging. Now, why is this important? This is important because when we are trying to compare test and reference product, it would be preferable to compare the products of the API in its native stage without the need for tagging of the API molecule in an effort to strengthen the observed signal from the molecule, which is necessary in some cases because the skin is a very heterogeneous organ. There are a lot of endogenous molecules that emit signal in the Raman within the Raman spectra, and therefore in some instances it is challenging to detect an API uniquely when it's applied on the surface of the skin. So as we move towards more powerful techniques, as we are able to detect these molecules without any tagging, that provides us with a more powerful tool to develop these techniques moving forward. Moving on now to cutaneous pharmacokinetics. This is another example from our GDUFA regulatory science research project, where we used Raman techniques to look at the cutaneous pharmacokinetics of these acyclovir products on the skin. So we applied the product and we used confocal Raman to look at the intensity over depth profile. And the data showed us that when we looked at these Zovirax products over time at 4 hours and at 24 hours, the amount of, or the concentration or the intensity that we observed from this acyclovir signal at 24 hours was much higher compared to this 4 hours across all the depths that we looked at. We then generated this data set for all the products that Dr. Rana showed you the data for, such as the IVPT data, and we correlated this data with the IVPT example. And what I would like to draw your attention to is within the Raman, using the Raman technique, we were able to illustrate that the bioavailability from the US reference product or the Zovirax product is significantly higher compared to the Austrian generic products at both 24 hours as well as 4 hours. And this data is very tightly correlated with the IVPT data sets as well as the DOFM data sets, which illustrated that the bioavailability from the US Zovirax product is significantly higher compared to these Austrian generics. So what we have here is a powerful tool which appears to be a sensitive and discriminatory tool that we can utilize for evaluation of bioequivalence. However, we are in the very early stages of this of utilizing or developing this method, and of course there are limitations. There are limitations related to detection of molecule in the skin. I already talked about some of them. There are challenges related to signal attenuation. I'll talk about them on the next few slides. There are challenges related to the utility of a tool as a semi-quantitative tool. If you recall, we were looking at the intensity of the signal and not the concentration of the API on the previous slide, and that is a limitation of the tool right now. There are challenges related to utility of the tool because a lot of molecules cannot be detected in their native state in the skin itself. So we have to address these challenges using more powerful techniques. There are challenges related to data collection and analysis techniques due to the sheer volume of data. And lastly, of course, you have to, for any method, you have to develop validation strategies to be able to utilize this method in a regulatory setting. So to be able to resolve some of these questions that are ahead of us, we have two current funding opportunities, one with Professor Richard Guy at the University of Bath, and a second one with Professor Connor Evans at the Mass General Hospital to be able to resolve some of these current limitations. And over the subsequent few slides, I'm going to show you how we are approaching some of these issues. So beginning with the detection of the molecule, the very first thing that we do is we take a spectra of the skin itself and we try to identify a region of the skin where the skin itself is spectroscopically silent. Now we look at the spectra from the molecule. In this case, we have cyanophenol and crisaborole. Both of these molecules have this unique carbon-nitrogen bond, which produces a signal in the spectroscopically silent region of the skin. This enables us to use then the area under the curve of this peak or the height of this peak to quantify the amount of cyanophenol that is present across the different layers of the skin. Now, how do we resolve the issue related to, now how do we utilize this technique? So we have to solve a problem related to signal attenuation. And what I mean by that is when you focus a laser on the skin, you see a gradual loss of signal or dissipation of the signal as the laser travels through the depth of the skin tissue. And this happens when a laser travels in any environment that has a high water content, which is also true for the epidermal and the dermal layers of the skin. Now, how do we resolve this issue? We utilized we utilize a signal from amide 1 bond, which is inherent to the structure of the skin, and we track this amide 1 bond across our depth of interest within the skin to ensure that the signal is relatively constant. Now we looked at the amide 1 signal or the decay in the amide 1 signal due to signal attenuation in a top-down experiment, and we were able to establish a log linear relationship between the amide 1 signal and the depth in the skin. Now, having this tool enables us now to correct the data or to correct for the loss in signal within the skin and obtain measurement, appropriate measurement of the concentration in any given layer of the skin. And what do I mean by that? We looked at top-down experiments, and I would like to draw your attention to the graph on the left first. So we used a saturated solution of 50-50 propylene glycol, and we tracked the intensity of cyanophenol across skin. Now, of course, you see attenuation of the signal due to what I explained on the previous slide. So we used the amide signal to correct for the signal attenuation, and we obtained a relatively constant intensity or what would be concentration of cyanophenol across the depth of the skin which we were looking at, so 400 micron. We then did a cross-sectional experiments, and what I mean by that is you basically take a piece of skin and you turn it on its side and you are looking at the concentration across the different depths, so at 100 micron, at 200 micron, at 300 micron, at 400 micron from the side which takes away this entire issue with dissipation of signal. And this is the data that is shown in green here. If you focus on the data now, what it shows us is once we corrected the signal for attenuation, the values that we got for the intensity of cyanophenol in the skin are actually true representations of the concentration of cyanophenol as it's shown and it's verified using these cross-sectional experiments. Now, how do we know that this method is sensitive to the cyanophenol concentration in the skin? It may be that the laser only always shows us that the intensity from application of cyanophenol on the skin is 30,000. Well, we looked at a 25% saturated solution and we saw that the signal also dropped to 1/4, where we obtained a maximum intensity of around 7,500. We also looked at yet another formulation, which is a different concentration of propylene glycol and water. It's a saturated solution, but again, this data was able to illustrate that when you have differences in the amount of the API in the different layers of the skin, due to differences in formulation, the method is sensitive enough to be able to pick up these differences. This is important because when we are trying to compare two formulations, a test and reference formulation, we have to be able to ensure that the method is sensitive and discriminatory to be able to pick up differences in relative bioavailability from two formulations when such differences exist. Last thing that I would like to show you is how do we utilize this for pharmacokinetic analysis. Now again, using the saturated solution of 50% propylene glycol water, we looked at the concentration of cyanophenol following 6 hours of drug application, and then we tracked the cyanophenol concentration over the entire elimination profile. And this is very similar to if we think about a solid oral dosage form bioequivalence study, when we not only look at the absorption profile, but we also look at the elimination profile over time. Now, how did we utilize this data? This is the uncorrected data and the corrected data. I'm not going to go into the details here. But if I focus on the pharmacokinetic profile, now we are looking at intensity over time and not intensity over depth. We see that as the molecule over time, the molecule gradually cleared out of the skin. And we were able to use this Raman-based strategy to detect the relative to detect the elimination profile of the molecule from skin. Now, once we replicated this data set for the saturated solution and the 25% saturated solution, you are then able to see differences between AUC and Jmax and parameters that are commonly used for comparison of pharmacokinetic profiles, enabling us then to utilize this technique for evaluation of not only bioavailability, but also evaluation of bioequivalence. The last thing that I want to show you is data collection and analysis tools. Now, this is an example from our collaborators lab at Mass General, where the authors use simulated Raman scattering techniques to look at distribution of the drug in different areas of the skin, so lipid-rich areas and water-rich areas. And the reason they were able to do this is not only did they use SRS, they also used CARS or coherent anti-Stokes Raman to obtain an image of the structure of the skin. In this specific example, they are looking at stratum corneum and the keratinocytes and the lipid distribution in the stratum corneum, and then they used deep learning and artificial intelligence to train their image analysis tools to be automatically pick up the structure of the skin and the relative distribution of the drug across the structure of the skin, which then results in a plot where you are able to look at the concentration of the drug in the lipid-rich areas compared to the water-rich areas. Why is this important? This is important because such automated image analysis tools will enable us to not only utilize this technique for detection of the drug, but helps us to efficiently process the images and institute adequate controls so that we are able to utilize these techniques in a regulatory setting. So with that, in summary, I hope I was able to illustrate to you that imaging-based techniques are not new to the pharmaceutical industry. They are a very important part of our analytical toolkit already. However, we see an incredible opportunity ahead of us to be able to use these techniques to monitor drug microstructure better, to monitor drug release, and for evaluation of pharmacokinetics. And to be able to develop this as a regulatory tool over the next few years, we want to identify strategies to enhance the applicability of this technique to a wide range of molecules, to be able to develop handheld tools so that we can use this in a pharmacokinetic study, to automate the data collection and analysis techniques, and of course, to develop method validation strategies so we are able to use this for evaluation of bioequivalence. With that, I hope, based on my talk today, the challenge question I have for you is, Raman-based techniques can be utilized to monitor what? They can be utilized to monitor the changes in the color of the skin. Does the skin look more red to you? They can be used to identify the indication for the drug product. They can be used to track a molecule that generates a Raman signal, or last, to understand the mechanism of action of the drug. Well, the answer is they can be used to track a molecule that generates a Raman signal. With that, I would like to acknowledge my entire team within the Office of Research and Standards, as well as all our research collaborators. And I would like to leave you with a closing thought from Dr. C.V. Raman, who of course received the Nobel Prize in Physics for his contribution and discovery of the Raman-based technique. The goal for us over the next few years is to be able to develop and utilize this technique for facilitating generic drug development. Thank you.